Hello, and thank you to the organizers for the opportunity to show how we use pharmacogenomics to target breast cancer treatments. My name is Deena Gendoo. I'm an associate professor in computational biology as part of the Institute of Cancer and Genomic Sciences at the University of Birmingham in the UK. Focuses on two main axes towards addressing disease therapies. One of them is really looking at how we can characterize preclinical disease models to efficiently profile disease. So, in other words, is there a way to identify what is really the right model that can recapitulate the molecular genotypes of the patient and of disease? And sort of tangential to that is trying to look at identifying what is the right drug that can really target cancers or target different disease subtypes and use those drugs for efficient treatment. And really, to answer some of these questions, which are in some cases very open, use a lot of different types of data sets. So the lab really relies on looking at a variety of different omics, multi-omics platforms, using integrated methods and machine learning to try and really compare different patient samples to each other, to xenografts, to organoids, and in other instances, looking at pharmacogenomics data and structural bioinformatics data as well to try and identify how drugs behave. And really, for the purpose of the presentation today, a lot of the focus is on pharmacogenomics, which plays quite a significant role in the work that we're doing. So, with pharmacogenomics data before, basically, this is data that looks at cancer cell lines. So we have a large range of cancer cell lines. They get treated by various drugs, and then basically we can profile these so that we can look at what we call drug perturbation profiling and drug sensitivity profiling. Very briefly, to break these down, if you're looking at drug sensitivity profiling, you have the cancer cell lines and we're treated with drugs at varying concentrations, and then we can measure the drug viability in the cell lines as a function of these concentrations. So usually we can extract what we call drug dose response curves, and we can pull out different metrics like the IC50 or the area under the curve or the area above the curve. And that for the work that I'm showing here today, we've been using a data set called the NCI60, which is a panel of around 60 cancer cell lines that were treated by over 5,000 drugs. If we're looking at drug perturbation profiling, it's slightly a different story. We're looking at the same principle that we have cancer cell lines. They're treated with varying drugs as well. But in this case, the cancer cell lines are transcriptionally profiled before and after the drug treatment. So actually, we're able to go back and really assess how genes are up or down regulated between those two states. So, in other words, what is the actual influence of a drug in perturbing the genetic behavior? So we used the L1000 dataset, which was released by the Broad a couple of years ago. This has a large sort of panel of cancer cell lines and over 20,000 drugs. So there's really a lot of room to investigate some of the drug behaviors in the context of the L1000 gene panel. What we end up doing is, as I mentioned earlier, we have the transcriptional profiles for the cancer cell lines before and after drug treatment. So we can actually use a linear regression model that allows us to compute what we call the drug perturbation. So in that case, we're actually extracting what is the genetic change that's happening basically from the cancer cell line we induced by a drug, irrespective of batch or concentrations or cell line identity. So I call this sort of a generic drug signature. So it's really just allowing us to see across all these cancer cell lines what is the influence of the drug on genetics. All of these data sets that we have, then we really have a lot of room to do some very interesting science and investigations given the richness of these data. So what we started to do is we started to investigate how we can use integrated pharmacogenomics to target some key pathways in cancer. And in particular, really the idea was, can we identify drugs that can target specific biological or metabolic pathways? So we started with focusing on the mevalonate pathway as a starting point in breast cancer because it presented quite an interesting challenge. In essence, the mevalonate pathway is producing cholesterol and isoprenoids, and so it's really one of those very key characteristic pathways that are helping in driving breast cancer progression. And aberrant activation of this pathway is a hallmark of many cancer types, and that also includes triple negative breast cancer. So if you block the mevalonate pathway with some compounds that are called statins, these are FDA-approved cholesterol-lowering drugs, but if you block the mevalonate, the MVA pathway with statins, that will actually produce a feedback response. And in essence, that would dampen the pro-apoptotic activity of statins significantly. So to get around this, statins would synergize with another drug called dipridamole, which together now would actually end up inducing apoptosis. And so seemingly that sort of solves the problem. Where's the issue? The issue is that if you're looking at dipridamole as a drug, it has a lot going against it in the essence that first off, dipridamole is one of those drugs that has non-specific binding. So it has a lot of polypharmacology. It's a non-specific inhibitor to the rate limiting enzyme of the pathway that we're looking at here, which is SRAB2. And also it has antiplatelet activity that precludes its use in some cancer types. So the question became, really, there's this pharmacogenomic need that's arising. We're trying to identify some potent inhibitors that can target the rate-limiting enzyme of the mevalonate pathway, but also the requirement is that these new drugs can also effectively synergize with statin and together really potentiate anti-cancer activity and target the mevalonate pathway in particular. So given all of that, the question was, can we identify synergistic statin-compound combinations now that can potentiate statin-induced tumor cell death in breast cancer? So we took this on, and one approach that we've been using is really combining a variety of different types of data. So we look at drug structures, we look at drug sensitivity, and we look at drug perturbation data. So with each of these types of data, we can actually take all the drugs that we have and sort of convert the data into a network of drug and drug relationships. So looking at drug structures, we can convert this to a similarity matrix that indicates how the drugs are related to each other based on their chemical structures. We can derive a drug-drug network looking at sensitivity profiles and responses based on IC50 scores. And with drug perturbation, we can also take that sort of matrix that we have of drugs and gene expression and generate a similarity matrix of drug relationships based on how genes are perturbed by the drug. And using each of these taxonomies, we use a method called similarity network fusion, which really fuses the three networks together and allows us to generate this very comprehensive network of drug-drug relationships across these different data. But now where the addition comes in is we are really trying to target the mevalonate pathway. So to really address this in particular, we can manipulate and we can change the drug perturbation layer that we're only restricting it to those sets of genes that are part of the mevalonate pathway in specific. And in doing so, we now regenerate a very pathway-centric drug network that we can take further and start to probe for drugs of interest. So in this particular example, we're now restricting our drug perturbation to all the genes of the mevalonate pathway. That generates a very specific mevalonate-focused network, and we can use that to start and assess drug analogs for our drug of interest, which is dipridamole. And using some permutation testing, we can now start to identify drug analogs that behave similarly to dipridamole, but also are able to target the mevalonate. In this approach, we've managed to identify 19 candidate drugs that share significant similarity to dipridamole. These span a diverse chemical and biological space. Some of them, such as doxorubicin, were already validated and shown in the literature for the synergistic activity with statins in ovarian cells. So that also gave a lot of credence that we're sort of addressing this in the right direction. Given the nature of the algorithm that we're using now, in essence, we're combining three different types of data. We can go back and probe our final network of drugs and assess between any two nodes, so between dipridamole and our query drug, any possible dipridamole analog, what is the actual data type that's been driving that relationship. And in doing so, we went back, we reinvestigated for our top 19 drugs, what was the driving factor. And we can easily see that manipulating the perturbation layer played quite a huge role in actually assigning these drugs as dipridamole analogs. We can go and now look at the drug perturbation data set itself and assess for all the genes that are the mevalonate pathway genes, what are the genetic changes that are happening across our drug of interest, which is dipridamole, and across all these new DP-like candidate drugs. And we can see that they share very similar expression profiles for the gene. So taking this further, really spearheaded by Dr. Linda Penn at the UHN and her PhD student Jenna Van Leeuwen, the Penn Lab ran a large range of experiments to assess the effect of these query drugs in targeting the mevalonate pathway, and really also assessing how these drugs would behave with respect to statin exposure. Would they mechanistically block the SRABP2 rate limiting enzyme of the mevalonate pathway? And what's the synergistic behavior? And from the methods that we'd identified a range of drugs, but I'm focusing here on two of our top candidates, which were really honokiol and nelfinavir. And in a second sort of secondary screen, we also identified two further drugs that I'm going to be mentioning a bit further. So using this approach, we were able to actually experimentally validate that these drugs do with dipridamole. And so this is just one very quick experiment showing those synergistic statin-compound combinations, looking at honokiol and which is HNK here, and looking at nelfinavir, which is NVF. And we can pretty much, this is a 5-day cytotoxicity assay to assess the fluvastatin and combination of fluvastatin with each of these compounds in terms of efficiency. And so we can actually see that very similar to dipridamole, if you play with the different drug combinations, there's also a dose-dependent increase in fluvastatin area above the curve value. So the compounds do potentiate statin activity. And using all of these findings, we're able to take it a bit further to evaluate statin-compound synergy using a Bliss index model across the cell lines. And that also shows that there's basically the same trend in synergy that we see between fluvastatin and dipridamole. We can also readily see with fluvastatin, nelfinavir, and fluvastatin honokiol. We got asked a very interesting question as to whether our algorithm would pretty much converge upon itself, or if we iteratively apply the algorithm now using different drugs. So if we were to use our drug hits as the query drugs themselves, do we convert to better drugs? And so actually we've been able to demonstrate that yes, if we do take our top hits and use those as the query drug instead of dipridamole, we end up cross-calling back many of those other hits in our list. So that really shows that our approach helps converge these pathways. So to recap, in essence, we had this problem. We knew about statins and dipridamole. Together they were working, but they're at a suboptimal sort of level. But by using pharmacogenomics and really targeting a specific pathway, we've been able to identify a set of pathway-specific analogs that we now know can synergize with statin towards better drug treatment. So this is really a good proof of concept of how we can take. We've expanded this pathway-centric framework with the help of two really talented trainees that were in my lab, Chris and Arminas. We're expanding this to look at a range of different drug signatures across tissues and across cell lines and across a large number of pathways. And this allows us to regenerate a lot of pathway-centric networks and really conduct over 200,000 possible different searches for identifying drug analogs against the drugs of interest. With that, I'd really like to thank members of the Gendoo Lab, in particular Chris and Arminas, who are taking this forward, as well as Dr. Marco Saponaro and his student Martina are helping with some of the experimental validation. A very big thank you to Dr. Linda Penn's lab at the UHN, who's helped a lot with the experimental validation. Initial phases of this work, looking at the SNF algorithm and developing drug networks, was with the help of Gary Bader's lab and Anna Goldenberg at SickKids. And these are all my sources of funding, and I'm happy to be contacted for this.